Hello, and thank you for joining us today for this roundtable discussion on removing quality roadblocks for cell and gene therapies, brought to you by Pink Sheet and sponsored by Lachman Consultants. My name is Maria Gerard, Managing Editor at Citeline, and I'll be your host today. Before we jump into the panel discussion, let's just go over some quick housekeeping items. First, if at any time you are having technical difficulties, click on the Test Your Connection button, and this will take you to a connection checker and FAQs page. If you are experiencing any technical issues, there is also a Chat Now function in order to go through to our support team who can assist you. If you want to enlarge the presentation, just click on the Maximize icon or drag the lower right-hand corner of the presentation window to enlarge. There will be the opportunity for Q&A at the end of the discussion today, so if you have any questions, we want to hear from you. To ask any of our panelists a question, simply type your question into the question window located on the bottom of your screen and then hit submit. We'll be answering as many questions as possible during this Q&A, and if there are extras, we'll try to answer you after the webinar via email. Because we know you'll want to listen to this discussion again, today's session is being recorded and will be available to view on demand in around 48 hours. I would now like to introduce our moderator for today's discussion, Sharda Millington, who's a consultant at Citeline. Sharda specialises in cell and gene therapy, working with biotech and pharmaceutical companies conducting competitive research and strategic market analysis. Sharda is also active in thought leadership creation, having published articles for Citeline's in vivo publication, as well as quarterly reports on the gene, cell, and RNA landscape, published as part of a continuing partnership with the American Society of Gene and Cell Therapy. And with that, I will hand things over. Sharda, the floor is yours. We may be having some slight technical difficulties, so maybe while we wait for Sharda to reconnect, we can start with some panelist intros. Keith, you are the first person I can see on my screen, so maybe I can come to you first.
Hello everybody and welcome to the session today. My name is Keith Lamb and I work for Lachman Consulting. I lead the cell and gene therapy and biologics practice for Lachman. I have about 30 years experience working in pharma and biotech, working across all platforms and all stages of development from early stage through to development and commercialization.
Brilliant. Thanks, Sharda. We were just running through some intros, so maybe, Richard, we can come to you next.
Hello, hi everybody. Welcome to today's event. Happy to be here. My name is Richard Dennis. I am employed at PPD. I've been here for about 6 years now. I work within our strategic development consultancy function as part of PPD, which is a global contract development organization, operates clinical trials. We're also now part of the wider Thermo Fisher group. We were acquired by Thermo Fisher last year. That gives us really strong access to manufacture for cell and gene therapies as well. I have about 30 years in the industry in biopharmaceutical development and manufacture. This is actually hands-on on the bench and from a regulatory perspective, also for cell and gene therapies as well. Really happy to be here today and along with my esteemed colleagues here as well. Thank you.
Sarah, maybe you next.
Hi, I'm Sarah Thomas. I'm head of quality for Regenix Bio. Regenix Bio is a gene therapy company developing AAV products for treatment of both rare diseases and larger indications. We have programs in early through late phase clinical trials. We have in-house manufacturing as well as doing some work with CDMOs. And I have been working in this business for 35 years now.
Fabulous. And Susan.
Hi, my name is Susan McClatchy, and I am the VP of Quality for Eterna Therapeutics. I've been working in industry for about 30 years, primarily in biologics and some medical device. For the last 16 years, I've been designing quality management systems for clinical stage companies to support in-house manufacturing of cell therapies. And thank you for inviting me.
And Sharda, I'll hand it over to you to get started on the discussion.
Great, thank you so much. Can you hear me okay?
We can indeed.
Okay, great. So yes, it's great to get the discussion kicked off. And I think before we sort of delve into the details of success factors and hurdles when it comes to sort of designing and implementing sort of an effective cell and gene therapy quality system, it would actually be really interesting to hear from the panel about exactly how you would define an effective cell and gene therapy quality system. So sort of what characteristics would it have to have? What sort of tests would it have to pass to be deemed effective? So it's an open question, but I suppose we could start with perhaps you, Richard.
Yeah, hi, thanks, Shada. So I think typically if we think about quality systems, if we're thinking of cell and gene therapy, a lot of cell and gene therapy emanates out of universities and hospitals still. And many of those might choose, for example, to build their own sort of GMP manufacturing facility there as well as part of that. So I think the first steps in putting a quality system together is see exactly who you have actually available. Normally for a very large pharmaceutical company, we have many people and we can pin positions onto different people. But within a small university, for example, we might have the actual originating idea behind this and a couple of postdocs maybe. So it can be very, very small in that respect. Universities seem to be better sort of disposed for that, certainly for gene therapies, cell therapy, and cell-based gene therapies. I think within that a quality manual is very good to start with that, and also the FACT and JC. We have very good available guidelines available for that. So FACT and JC have a very good, and again, it's more from the cellular side of things, have a very good quality section as part of their paperwork as well. Also, ICHQ-10 is a very good global sort of starting point that clearly lists out the responsibilities in terms of your documentation systems, your responsibilities in terms of personnel, etc., and your responsibilities in terms of your facility. And I'll hand across to my other colleagues to speak as well.
Oh, you're on mute, Dada.
Sorry, I was just handing over to Sarah, perhaps.
Sure. So, given the complex nature of cell and gene therapy products, in my view, it's really important for quality systems to be flexible in finding ways to meet regulatory requirements, because we do often have to make adaptations to traditional approaches or develop alternatives. Systems really have to focus on identifying risks to product quality and managing that, again, because we can't always fit the traditional molds. So we have to consider what the risks are for these alternative approaches we may need to take. As Richard mentioned, we're often working with small organizations, so it's really important that systems are streamlined so that they can be reproducibly executed by those small teams. You can't afford to have a big complex system that a small group can't manage. We have to think broadly in scope, covering all the way from starting materials to delivery to individual patients. And of course, as in a good quality system, you have to collect data, learn from it, and plan to continuously improve in a controlled way.
Amazing, thank you. Keith, Susan, is there anything you'd like to add to sort of your perspective on what exactly an effective quality system consists of?
Yeah, just a couple of comments to make there as well, Shada. I think I'm definitely from the school of looking for, is it appropriate for the stage of development? And that's usually the starting point for considering where you are with your quality system and what you'd need and what we'd advise to put in place. And then really, I tend to start looking at, what is in place, how it's used, and then, in what degree can you determine it's effective? And there's a whole host of onions around that assessment process. But I think particularly for cell and gene therapy, I'll reiterate something that Sarah said. Really, the risk management requirement is so important, and to really demonstrate you've understood the risks that may be there, there may be risks you're evaluating for the product, and put that in context of what's known and uncertain about the product, and then how the quality system fits together. Again, reiterating the particular type of a facility and environment that the manufacturing organization is established in, and then managing change. I think the management of change associated with identifying risk has got to be central to your quality management system for it to be effective. And then in today's modern world, really, and probably because the added risks around particularly autologous products, but data integrity and how data is governed within your quality system, it's so critical for patients, but it's so critical for the data that you manage and ultimately the final applications that you're going to submit.
And I have to agree with Sarah about flexibility. I think that is so critical, that your system has to be flexible enough to support the number of changes that are going to occur over your drug development process. It may be a change to the formulation, how it's administered. The company may change from in-house manufacturing to outsourced. You may change your targeted clinical countries. So the system has to be flexible enough to support the change without requiring that the quality system itself be changed. And in order to do that, you really have to have a robust change management system that isn't going to hinder rapid adoption of process improvements of these changes. Flexibility is just, I think, the key.
That's great. Thanks so much. And I think you raised loads of points, which a bit later in the discussion, we'll talk about hurdles. And I guess one of the biggest ones is kind of reconciling standardization with flexibility. So it would be great to explore that in a little bit more detail. But first, so you've kind of gone through what constitutes an effective cell and gene therapy quality system, but it would be good to know what kind of considerations or what steps need to be taken in order to sort of successfully implement one. What do developers need to keep in mind in order to actually get there and achieve a successful and effective cell and gene therapy quality system? So perhaps could we start with Keith on that?
Thanks, Shada. Yeah, I mean, I think the steps you take are understanding your product in your environment and understanding the system that you're trying to control and the stage of development. But I think your starting point always has to be consideration of culture and an objective that's collective around the organization. As you set off on that journey, and very often, as we've heard, even in this early part of the discussion today, you know, the role that's played in academic and research environments still in the emerging cell and gene therapy space. And that movement and that commitment to change from a very academic or R&D based organization to one that is going to put some structure and some restrictions as it may be seen around people, but do that in an agile and adaptive way. And so I think the starting point is setting a clear objective and a commitment to understand the cultural change and manage that change. And then the mechanism of putting in place, you know, your quality policies, your individual quality systems, and choosing that and tuning those to the particular requirements of cell and gene therapy becomes a design and implementation phase. Whether you're moving to a cell and gene therapy CMO who probably has an established quality system and understanding how that works for you, and you will put your oversight of that in place, or you're developing your own system to implement internal manufacture, which many companies are continuing to do because of the state of capacity in the industry. Those are probably the starting points to kick it off.
Great, thank you. Richard, I see you've got your mic off. Do you have anything you'd like to add on to that?
That's a good sign. So hi, the great points that Keith's made there. I think really to add to that, these are a bit of a case study. So once I worked out in China for a university-based, a hospital-linked university-based facility. They just had a new GMP unit built, but that was it. They had the money, they had the resource behind that, but that was it. They didn't know where to go from that. And they said, could you come out and just help us point us in the right direction for putting a quality system in place? I think, again, as we've collectively said, this is very much risk-based in terms of cell and gene therapies. It has to be all the way through. So it's working. You need to build this from the ground up. So you need to see what currently is in place. And from the university, that's normally sort of fairly basic, or can be fairly basic. It meets with the university parts. From a hospital perspective, it could be at a sort of slightly higher level there in terms of its operations. But really looking at it again, maybe somebody who's done that before. So somebody either in your company at that time, the companies can be quite small, but people that have set up a quality-based system before, or to get in a consultant who's done that, can save you a lot of time actually piecing that together. I mentioned about the guidances earlier. The guidances are very helpful, but we've got to be careful. And we've mentioned about a stage of development as well, where we're at. So we're starting from the ground up. We don't want to be able to put a fully blown quality system straight off in the space of about a day. That's just not possible. So we need to work with what we've got. As I mentioned, the FACT and JC guidelines are actually quite good. They give a listing of what's required within a quality system. So they're great to just literally print off. They're free online, as long with ICHQ 10, for example, as well. But that's fully blown. So you just need to pick out the points that are efficient for you as well as the ATMP guidelines as they relate to GMPs. Also have quality system sections as well. So amalgamate all of the information that you can. Start listing out what those headings are and start filling in what have we got here, what haven't we got, and then you can do effectively your own gap analysis of what you need to be able to fulfill and put in place.
Great, thank you. So it sounds like you need a very structured plan right from the very, very beginning. I'm just kind of wondering, Sarah, maybe you could touch on this, but what kinds of supply chain considerations do you need to have in mind when you're sort of constructing a successful quality system?
Yeah, sure. So, I mean, supply chain in cell and gene therapy products is more compressed, actually, I think, than in a lot of traditional therapies, in that we have to consider manufacture of these starting materials that even feed into making the API, and then we go all the way to delivering to individual patients. And so, you're thinking about a delivery system that may actually be almost more like a clinical trial approach rather than commercial in a lot of ways, because you are, going to an individual patient even for commercial delivery a lot of times. So your supply chain, I think, is longer. In terms of, starting materials, we're often working with unique things, and we may be working with single sources, at least early on, so that can be a challenge. We also use a lot of disposable systems, and so that adds more materials that you have to control for, and things that can be difficult to get and in short supply a lot of times. So you have to really plan very far in advance and think about contingency planning as well to deal with supply shortages. Another issue, I think, is that for a lot of smaller companies, if you're working with larger suppliers for, say, some of the more traditional materials or the big plastic items, they don't always want to give you the time of day because you just don't provide enough volume of business to them. And so it can be really a challenge to get, permission to do an audit or to get them to respond to quality questions. So you have to start thinking about what other options do you have. If you're working with a CDMO, do they have more leverage? Can you use third-party audit groups, for example, to purchase an audit, or, focus on more risk assessment and incoming controls if you really have a hard time. So it's, more complicated. So lots of things to consider in terms of the supply chain.
Great, thank you. Susan, just interested to hear your thoughts on the kind of considerations developers need to have when trying to construct a successful and effective quality system for cell and gene therapy.
Well, I think it's important to understand, first of all, your product very well and what your compliance is going to look like, where clinical trials are going to be performed, and structuring the system so that it can support, again, rapid change. So you would start with a basic core system, and I would recommend designing it so that it can accommodate for both in-house manufacturing and if the company decides to outsource without having to change your whole system. And then have it be a kind of a modular platform where, as you're going you have a solid plan for how you're going to implement higher levels of GMP as you're going through the development phases, modularize things so that you can add on things as they become needed, say validation. Now you've got your set of procedures that you can put in place that will handle validation. So I think that it's being flexible and being able to add things on that will help to get that system in place.
And Shada, just one further comment on that. I think the pace, when we work in the CMC space, we're often trying to stay off critical paths and ensure that the clinical programs can move ahead. And I think the quality system has to run in parallel with your development program, your CMC development program, to keep pace with the aspirational clinical timelines. And if you don't do that, you are, so sure to run into further delays down the line. So again, coming back to thinking about change management and assessing your risks, that also has to be, in terms of the adequacy and completeness of your quality system. And I think that important aspect of keeping pace with the clinical program and staying ahead of that is critically important.
Great, thank you. It seems like risk management is kind of a big buzz topic. That's obviously a very important consideration for these quality systems. And it would be great to sort of move on to talk about the hurdles when it comes to implementing and creating an effective quality system for cell and gene therapies. So perhaps we could stay on that topic of risk management. I was just sort of wondering, what are the sort of blocks that developers could come across here? What are the sort of dangers that could arise there when it comes to risk management and risk management considerations?
Susan, I'll take a stab at that. I think that risk management is being, in recent years, as we've really started incorporating that into all aspects of the development, how you deal with investigations, all within your quality management system. And I think that all of us inherently as quality professionals do the risk assessment in our heads. Anytime we're dealing with any issue, we're doing it up here. But what sometimes is behind is documenting it, make sure it's done in a formal way, and have a kind of decision log where you're keeping track of, you not only have your risk assessments, but keeping track of why you made certain decisions along the development process, because they may have made perfect sense to you at the time, and years later, you're trying to remember, why did we do that? So I think just the documentation of risk management, and not locking yourself into thinking that means I do an FMEA, because there are a lot of different ways to analyze risk. And I don't think that the company should lock themselves into one way. They should be flexible enough to do what fits the situation.
Great. Did anyone else in the panel have any thing they wanted to raise on that topic of risk management and the hurdles that people could come across?
I'll follow on from Susan, and I think within that as well, we've got to consider that our first stage is moving products from the bench through the first into human. If we're looking at putting together a system, and there's a lot behind that, so we're a new startup, we're a new biotech company, we've just got a first stage investment as well. If I say to the investors, look, we're going to build our own in-house GMP unit, we're going to do that and go forward, investors might say, right, okay, show me how you're going to do that, but fine. If I said, I'm actually going to go with a CDMO, going to go with an outsourced partner to do that, it's going to be more costly. But the investor will probably say, yeah, that's fantastic, because they're tried and tested, they do this day out and day out. And CDMOs have become much better at doing that. So that's something to sort of really evaluate as part of your overall risk programming and assessment and strategy as part of that. I think things that can typically pick you up is that as a fledgling emerging biotech company, great, we got an investment, let's go. I think one of the dangers behind is that these aren't small molecules as well. So there can be certain resources involved in the development of these projects that aren't aware of the complexities of cell and gene therapies and in fact that we need to use a risk-based approach for those. You can't compress, moving on, speaking to Keith's point as well, we can't really compress a CMC. And that's the issue that we've got. It's one of the safety factors that we need to build in place. So we need to have all the correct safety testing in place as part of that. That's very critical. So I think the main hurdles in place for me is the fact that the product, as we said, is absolutely focal to what we're doing. We can't lose sight of that. We really need to link into into non-clinical and clinical. Those joined up parts need to certainly be there. And as a very small company, it can be quite difficult to get all of those things in place. So either good consultation is good, or somebody within your executive board already has that understanding is very critical to help drive that program forward. And mitigation early on is very key to not tripping over further down the line. And your biggest risk is the one you haven't thought about. So
yeah, you've touched on some really interesting. Oh, sorry, Sarah, did you want to jump in there?
Yeah, I actually just wanted to add on, marrying together the scientific risk versus the regulatory risk, and really making sure that, when you're taking these scientific or alternative approaches, that you're working together with your regulatory affairs team and seeking feedback from the regulators. A lot of us are working with, say, orphan diseases, rare diseases, where you may have the opportunity to have some additional interactions with the agency through something like the RMAT program, or even just like a routine type C feedback. So you can get that, discussion with the agency. And I think you'll find that when you've got a good scientific justification, the agency, will be, willing to work with you on alternative approaches, but you want to find that out early on. You don't want to, get halfway down the road and realize that they don't buy your approach.
And I totally concur with Sarah's comment there about engagement. And to me, I come back to culture very often, especially in the early phases, because an organization that really isn't adept at reaching out and engaging and asking for either expertise or dialogue with regulators, that openness and acceptance of putting the framework and structure in place, I think has to be the starting point to really get the company ready to establish a quality system, and then really disclose where the concerns are, be open about what the challenges are, because there are limitations, right? There's limited knowledge in product attributes, there's limited manufacturing history, there's huge variability in starting materials, and I think those factors are known to the regulators, and there are mechanisms in many regulators setups to reach out and talk about new technology and innovation, and those offices are absolutely encouraging that in the cell and gene therapy space. It's an opportunity to go speak with them on specific challenges.
Right. So you mentioned there are these sort of mechanisms in place when you're discussing at an early stage with regulators. Is there anything more you think the regulators could do to help developers at an earlier stage? Is there sort of additional steps that you think could be taken, or that would be helpful to see?
Certainly in the, if I might go first, it's certainly in the US, we have very good insight in terms of interact meetings that are very, very open. And also in the EU as well, very, very similar sort of fast track type of approaches, if that fits. But the interact meetings are designed for very early stage sort of companies to sort of really come in and speak about what they want to do, how they can sort of develop their sort of early stage sort of strategies. And this could be really when they're still on the bench at that sort of point, at that sort of feasibility in effect. It's not really until after that we start moving into sort of more programmed interactions with the agencies. Is where more hard and fast interactions. But those early stage interacts and similar EU meetings as well are very strong in providing that. I think we've got a very mature set of guidelines in place at the moment. I think I would encourage everybody to read those. I think we're all that we're inundated with guidelines left, right, and centre, and we can be overwhelmed with them. But it's really important to pick out those key ones, especially in the early phase. There's guidelines for investigational products, so they're the ones that literally pull off And just by having those couple of guidelines in place for cell and gene, they work tremendously. And also then speaking with the agencies early on, their scientists as well, they're very happy to engage. So I think they're doing a lot already. But that's something that we really need to, as an industry, really sort of come on board with and recognize that as well.
I think it's what you're mentioning about reaching out to reading what's already there. I have to mention the Alliance for Regenerative Medicine and A-Cell and A-Gene that came out. Those are just invaluable in helping companies structure a product development platform and quality by design and how you apply that to these type of products. So those resources are out there to really look at.
Yeah, and if I could
Hi,
add,
Sarah.
really participating in these industry groups. There's different consortia out there, and ARM was mentioned, Bioforum is another one. There's a number of Nimble, there's other groups out there. So it's really important as an industry that we engage and participate, because we can also influence what appears in some of these guidelines. The agencies publish drafts, and we have the opportunity to get feedback when we see things that, you know, we just don't think are going to work. And the agency has shown that they take the industry feedback. I mean, good example, they had a guideline on neurodegenerative diseases that had some information that industry largely didn't agree with, and the agency adapted. So it's important to keep that dialogue open.
Yeah, I think we're very much a self-regulated industry, so the industry does become very much part of that whole regulatory equation. So I agree, Sarah, with that.
I think recently the guidances, as we're talking about guidances that are available, the manufacturing change and comparability recently was published by FDA. So that's a great example, Sarah, for one where either working with industry bodies or feeding commentary back. I'd also give a plug to ISPE and PDA, the organizations which have some very excellent working groups that their conferences are fantastic to network and learn about application of quality systems.
I'm not sure Shada might be having some technical difficulties again. While we wait for her to get back online, though, just a reminder to the audience that we will have the opportunity for your questions towards the end of this panel discussion. So don't forget to submit those in the question box, which should be at the bottom of your screen underneath where you can see the panelists today. While we are waiting, though, we have already had one question come through, so I might open that up to the panel now. So that question is, if you are looking to have multiple sites with different quality management systems providing cell-based starting material or cell processing services, what are some tips for best managing this as a sponsor? So I don't know if any of the panelists want to take a stab at that. I can see we have Shada back, but maybe we can answer that question here in the meantime.
That's Steve, please.
I was going to say, I mean, it probably depends a little bit about whether those are internal or external facilities. I think if we were talking primarily an outsourced an external system, the criticality of getting a very effective quality agreement in place, quality and technical agreement, you know, separate to your commercial terms, you understanding the flow of information and the disclosure of issues and involving those partners in investigations that go back to those organizations. So it is about assessing those type of organizations, but it's also about building a kind of a quality management framework with them and a partnership with them. So, you know, you need to have an external management program that, you know, engages, isn't just sending documentations back and forth, but is totally intimate with the operations of those service providers for you. And obviously, the more critical those are, and you would achieve that by your effective risk management system, you can develop a tiered approach in terms of how you monitor, work with those suppliers and service providers. For your own network of internal service provision, I think there's often a debate with companies that are, you know, come from different histories. as to the pace of change and the pace of integration of your quality systems, but certainly alignment on key aspects of those in terms of timing and the actual process flow and how you assess data and how you integrate and work through your deviation to change management system to ultimately the flow of data that will support the disposition of a product. I think that integration of the different nodes, it can never be more important than it is in cell and gene therapy. I think for all of us who've worked in this space, the proximity to the patient and the patient's role in this with a clinical or a hospital center is so different. So, you know, the juxtaposition of a clinical operation against a GMP framework, you know, those interfaces are where you should spend your time managing and making sure that your quality systems align between the different nodes.
I think to add on to that, we work at very sort of different phases when we go through our various clinical phases, and certainly at that sort of stage 1, first in human, we're working again, most notably, most often with a smaller sort of biotech that could be linked in with a university. We're coming from the procurement stage. It's a cell-based therapy, so procurement's going to have another set of quality systems compared to the university. We may do an investigator sort of led trial as well, or may then and work with the CDMO for that sort of position. We may then want to move with a commercial orientated CDMO in that respect as well, that might have more concrete types of quality system in place as well. Aligning all of those is really key, and especially as it relates to the types of materials that we use, moving from research grade through to GMP grade as well. Making sure that it's written down. If it's not written down, it didn't happen. So all of those things, again, in aligning are really important. And even though we're very much sort of have restrictions on being able to move around at the moment, audits of these facilities, some of that can be done as a paperwork exercise, but physical audits are very important as well, actually going to the facilities, inspecting those sort of quality systems that they have in place as well. I think is very, very key to this.
And if I could add, I think also just making sure that there is a central point where everything is being pulled together and data is being analyzed, and manufacturing processes and experience is being analyzed across the multiple sites. And if there are lessons learned, that they're being applied from one site to another as well. So, just somebody looking at the bigger picture, making sure, as was noted, that everybody stays in alignment, and that the quality is maintained in a consistent way and the process is executed in a consistent way across all locations.
Great. Jada, we're just answering one audience question, so I'll hand it back to you for the remainder of the discussion.
Thank you. Sorry about that. Just before we talk about removing hurdles and ensuring patient access and speeding up processes, I did want to talk on, I know flexibility was highlighted very heavily, and I think that's an important thing to touch on. In particular, I'd kind of be interested to hear your thoughts on how you reconcile the need for flexibility. You've got to keep up with such a rapidly evolving area when it comes to cell and gene therapies. There's evolving regulatory requirements, movements into larger patient populations, new technologies coming out. So flexibility is extremely important, but how do you then reconcile that with the need for standardization and consistency, which is so important for sort of quality considerations? Keith,
sure.
Feel free
yeah,
to
sure.
get that.
No, very, very happy to start off. I think actually I think Sarah mentioned agility early on, and unfortunately, maybe people who look at from the outside and not from within a quality system don't often say it's agile and nimble. But I mean, that's got to be our aspiration, and particularly to make it work for the type of product and the urgency that we're trying to move through development to commercialize, to serve many, many patients. And I would say for me, it's really about how you organize yourselves and how organization is put in place with communication. And that has to be key to be able to connect. From my experience of working in the cell and gene therapy space, the connectivity and the communication to clinical perspective, to clinical physicians, particularly from the technical organizations, and of course quality is part of that. to make urgent and needed decisions on particular batches and particular patient situations, that communication and connectivity is absolutely key. And I think where we were successful, we had excellent connectivity and communication, and organized in a way that people knew exactly what decisions and what information needed to be made those decisions. And I think that does, I'm a very strong, firm believer that you can have high quality and compliance and still achieve exceptional speed and exceptional service to patients. And of course, from a quality background, we want to continue to improve and drive those changes. So being in tune to what maybe isn't working or what's getting stuck, and being agile enough to adapt, but all within a framework of an established quality system, it is absolutely deliverable.
Sorry, Shadi, you're on mute.
Sorry, I was just going to say, do any of the other panelists have anything they wanted to add to that?
I would add that the one thing that you can't do is say this is how it's always been done. I mean, that's that is the worst thing that I hear when I ask why is this is why it's how we've always done it. And the quality system, as we said, has to remain flexible, and we have to be able to incorporate lessons learned. And we're going to be learning them all the way along the process. How do we now get that feedback back into the system so that we can streamline it to be more supportive of the changes that are occurring?
And I think also to add to that, if we're looking at flexibility, we've got to move within the frame of the fact that the key factors that we need to show is product quality, so that's paramount, and also patient safety, that's another thing. And the two of them goes really hand in hand. Again, going back to the fundamentals of CMC, non-clinical and clinical, they need to work flawlessly together. So they need to act to be flexible very well together. And I think if you have any one of those sort of stages or any of those sort of subunits that falls over, then it's going to impact everything. It's going to hit back the clinical timeline. CMC is very much on that sort of clinical path as part of that, as is non-clinical. So we need to sort of build into all of those. Look at mitigating early as well. I think is a key one. Make sure that we have knowledgeable people in the mix as well. So if we do come across any issues, any quality issues, we can quickly look to resolve those points and move on. I think we're also in an industry where we have guidelines in place that sort of seem to be quite fixed, but we can push the boundaries of guidelines as well where that's necessary. Again, these are a risk-based approach for these types of products. So we've got to bear that in mind as well. Guidelines are guidelines. So I'm not saying, okay, let's not follow the guidelines whatsoever. Extremely important to do that as a backbone. But we can, again, if our system doesn't quite work with that, look at the logistical viewpoint behind that. And the agencies are very happy to listen to that as well, providing it's logical and still meets safety and still meets patient safety.
And I would add to build flexibility into your procedures. So it's not making it up every time, but you actually have procedures that define this is how we approach things. This is how we evaluate risk. These are our risk categories. This is how we break things down. So if you have it outlined in your procedures, you can actually, have that flexibility, but do it in a consistent way.
Right, thank you. And on the topic of standardization, I guess this is where potentially from a quality perspective, the thoughts of platform technologies it comes in and could be really important to discuss here. I don't know if anyone wants to touch on that and how important that can be to incorporate. Keith or Sarah?
Sure. I mean, I think I'm seeing and probably fully bought into. I think the future for the cell and gene therapy area for companies to be successful, based on the cost, the time, the uncertainty, the flexibility, is driving to platforms. And I think biopharma learned that after years and years of development, and many companies now have very standardized platforms. And I think if cell and gene therapy companies can learn to drive to platform earlier and as quickly as possible and establish platforms and not reinvent and slightly modify process, in development, and then really do think about the scalability approach. Is it scale out? Is it scale up for different type of products? And have that considered from a commercial mindset as early as possible. I think would be the two things that I would say on that, Shada, because it is likely that if you have a standardized platform, you'll be able to drive costs down. You'll probably be able to drive timelines down. And I think that's going to be critical for the viability, not just of your first product, but hopefully a platform of other products that follow through.
Great. Sarah, did you want to add?
Yeah, and I would just add, from a quality perspective, I'm a huge fan of a platform basis. Apart from, increasing your speed and efficiency, I think improves your reproducibility because you're doing the same thing or essentially the same thing over and over. Your operators get used to it. It reduces your training burden. You can use the same batch records to some degree. I just think helps, standardize your quality operations and improve, the ease of manufacturing in a consistent way.
Also going back about 10 years ago, this is quite a new area as well. We've got to consider that this is really sort of a learning ground for a lot of people. I think we're very valid, really sort of stepped forward quite a long way since then. If you're looking at any CDMOs at the moment, many of those have really now solid track records of a good several years and more in this arena. We've also got very good guidelines that are in place now that give way to having template processes. I've worked in the CDMO industry backgrounds. And with that, to actually validate the facility, we ran about 20 different processes through that, candidate processes, monoclonal antibodies, recombinants, different cell and gene therapies, which back in the early 2000s, they were very much in their infancy. But we still did that and still had a sort of a template process that we said, okay, if this is a cell therapy or gene therapy, it would roughly fit into that type of format. And I think we're in a much better position to do that now, and that's a good thing to take advantage of. And if we're developing purely in-house, then we've got to do the classical of beginning with the end in mind and knowing where you need to know where you go in order to be able to get there. So I think that's really important, and that's part of that, to be very objective and not empirical and say, okay, that didn't work, let's try this, okay, let's go here next, because that can really slow you down. So I think having those template processes, again, as Sarah said, that we can just lift off and use, that's the key way of getting forward for this. I think Peter Mark said from the FDA that this industry is a bit like sort of an aeroplane. It's been learning to fly each time in its early stages. But now moving forward, we know what an aeroplane is. We know that, so we can slot into that. And I think, as Keith said, we need to sort of put some drivers behind this to really make this work.
Fantastic. And Susan, did you have anything to add on on what your fellow panelists have said on that note?
I would just say about platform technologies, I think it's just important to reiterate to get quality involved early when you're developing these platforms. I think very often, especially in new companies, first product, the need for quality is not really thought about until the product we're ready to file an IND. Okay, we need a quality system. It really needs to, I think the earlier upstream you can move quality, you can head off some of the problems that might occur later on and have them working as this platform is being developed and how they're going to structure the quality system around it.
Fantastic. We do actually have another question in the Q&A inbox, so perhaps we can just take a moment to review that. So it is a little bit of a longer question. It says, within the regulatory context and discussion today, it'd be really interesting to get an understanding of the CTD organisation for cell therapy products, from DS, DP sections, donor cells, feeder cells. I don't know if anyone wants to take that on before we get into the final part of the conversation today.
I think that one might be a better question for a regulatory affairs person, to be honest. I mean, there's definitely a structure outlined for the CTD. It's, that's published. And so I don't think that that basic structure really changes in terms of product type, but to get into, nitty gritty details, I'm afraid you probably need to talk to a regulatory affairs person. Richard, you're on mute.
Obviously saying something very important. I'll take a stab at that, and I think really as Sarah said, we have very good guidance in place. We have the M4Q, which is the CTD structuring as part of that, and module 3 within that as well. Also, we have, again, guidance for investigational advanced therapies. This is more so from the EU, but I would say wherever you are in the world, whether you're US, APAC, EU, have a look at the guidances in the other areas. Again, EU and US are really easy ones to sort of dive into. And they have a lot of commonality between them as well. So if you're developing an investigational product, the EU has very good guidelines in place for advanced therapy investigational ATIMs, I think they're called. So for cell and gene therapy, that's good in that respect. The standard CTT guidelines also state what needs to go where. If we're looking at these types of products, and in terms of, again, sort of feeder cells and our cells in general, I think in there we need to understand what the feeder cells are, again, because they are effectively, if they're not adding to the mode of action, they are an impurity. So that's something that we need to consider. It's not saying that by avoiding feeder cells that's good, but sometimes we need to use those, but we may have some of those in our products. We want to eradicate those as much as we can, etc. To show that we have very good and firm control of our actual products as part of that. But within that, certainly within the sections of the CTD and within module 3, we'd look at that, placing those things. Certainly we'd have part of that in impurities for the feeder cells as well, how we've addressed those. We would look at that as part of our manufacturing development sections and as part of our characterization as well. But anything that's as part of that in terms of DS and DP, we also need a very strong demarcation between the drug products and the drug substance. For these types of products, sometimes they're more of a continual process. They don't really stop at drug substance. They continue running through to drug products. And I think as part of that, we still need to be able to put a line in and say, well, this is effectively our drug substance. It could be a case where we take our products all the way, our drug substance all the way through to the final sort of vialing, and we just call our final vial the drug product, for example. So we need proper demarcation of exactly where that is, and I think that's a very key point between those. But good guidance in place of where things should hang and slot, but there are sort of rough sections where we should place these parts.
Dara, I'll hand it back to you to conclude the conversation. We've answered another couple of audience questions there.
Okay, great. Thank you so much. Yeah, just in the final 10 minutes or so, would like to talk a bit more about actually removing the hurdles and getting these therapies dosed into patients a bit faster. So I know we sort of touched on this a bit briefly when we were talking about flexibility. But I think it's just a question of in what ways you think we can sort of speed up the process of therapy development, but while still maintaining the sort of high quality that regulators need to see, and to sort of fit all of the regulatory requirements. Perhaps, Susan, could we start with you?
I think having the product development plan overall in place, and I think sometimes in small companies, first product coming through, they don't have a defined system for product development. How are we going to handle tech transfer? How's that going to go into manufacturing? And having that system in place and defining what's needed at each phase to get through these phases can help speed it up tremendously.
Yeah, I mean, many companies have defined, ways of working of differential development and delayed development, and I think it's really acute to decide what things can be effectively delayed, what things can be built as a platform, and what things can be not delayed and are an absolutes, and maybe things that are impediments for moving to the next phase. And I think getting that decision making with really the risks around the product and the facility and the materials and the people and your growth curve of hiring people. There are many factors that can, you know, be the result that slows you down, that may not be the obvious one from the start. So, you know, for me, it comes back to that, as Susan said, a very effective development plan and an integrated plan that keeps pace from your CMC development with your clinical aspiration, and a very effective risk management program that genuinely assesses the risks to your program, as well as the risk elements within your the technical aspects of your product, within your own organization, and sourcing expertise to support the growth in the next phase. I think those are just perhaps done a little earlier is probably the strongest message I can give, because you know, you've got to manage change in a different way when it's a highly complex system that you're trying to change. And I think that's what the regulators are going to consider, and your risk of clinical holds and your risk of delays of applications. I think they're not all pricing related, right? They are often technically related from a commercialization. And we do see many clinical hold situations within these kind of therapies. So I think that's where you have to have a comprehensive integrated plan that's solidly based on very effective risk management that is focused on patient safety and the effect and impact on the quality of the product.
Yeah, and I would add that, you know, it's really important as part of that process development plan to consider what elements of your program or process you need to identify upfront and have established early. You know, something like your genetic construct, you don't want to change that, right? You want that set from the beginning, because that's going to impact your clinical performance. And so if you try to change something that fundamental, the agency is going to send you back to go, because the clinical studies that you've done aren't really going to support the intended product. So think early, you know, what are the things that you don't want to change that are going to be a problem to change later on? So make sure you establish those things early. And, you know, the agency actually the FDA put out some of their thoughts, I think, in the recent comparability guideline or draft guidance that they just put out, you know, talking about kinds of changes that they would not want to see made late in your clinical development. So look at the guidance that's available and just also logically think about, you know, what would be a significant change to your product that could impact the clinical trials and set you back.
And to add on to that, how do you evaluate the change when you've made a very small number of lots? You have maybe preliminary critical quality attributes or critical process parameters. You may not have a potency assay. How do you evaluate that change? And I think that's something that is so important to think about early, to set some basic guidelines around how you will develop or how you will evaluate any change in the process.
Excellent. So sort of ironing things out very early, thinking ahead, is extremely important. I know we've got a couple of minutes left. I just wanted to ask a question a bit about the sort of responsibility, where the responsibility lies in trying to get the balance between sort of high quality and speeding up the process. I just, out of curiosity, I mean, I know you mentioned consortiums, for example, earlier on in the discussion, and collaborations. Are there more that companies could do to work together to help each other out and sort of, you know, speeding up the process of getting a first patient dose when it comes to quality considerations? Does that work? I'm just thinking, you know, the idea of sort of being a bit more open source has often been floated, and just wondering what your thoughts are on that from a quality perspective. Susan or Sarah, maybe.
I mean, I think it's hugely beneficial when you can speak with, you know, folks who have maybe gone before you in terms of being a little more advanced or parallel with you, maybe facing some of the same issues. You know, really helps if we can learn from each other, as much as we're open to that. It, you know, gets a little challenging. Folks get, you know, a little protective of their intellectual property, but there's also, you know, more general things that I think we can feel free to share in terms of, what the agency is asking for in terms of specific types of analytical techniques or comparability approaches or whatever, that type of experience. And, as mentioned earlier too, the more we can work together to influence the agencies and the guidelines so that they are industry friendly, that's all hugely beneficial.
I think that
Charlotte, sorry, Susan, please.
I think as industry, we have more collective power than the agencies have, and so the weight is a little bit more on us to help each other along. And it's not something that you hear too much about publishing. You hear about publishing on technology and publishing on your studies, but maybe we could, in the realm of open source, publish lessons learned and things that we see that could help other companies with similar type products.
I think, Charlotte, this platform today has just proven, I think, how we can interact and answer very generally without getting into any intellectual property or anything like that, but what our thoughts, opinions are, what our experiences are, also to share with the audience. We're very much part of this in terms of some questions that they have as well. So I think more of these types of interactions. We mentioned earlier, again, about the various industry and sort of groups out there as well. Unfortunately, nowadays to get to conferences, it's quite expensive, but I think for early stage biotechs, it's still possible to do that, but you have to sort of really pick and choose. But some of these events, for example, and some of the events that are free online are very strong. And again, any of those that you can get to, I think today's a good testament to that. It can really provide some good, great, solid and collective answers.
Yeah, I think that's right, Richard. I think that their expertise is out there and it is available. And I think it's whether it's tapping it in a conference forum and networking, or whether it's in directly or hiring expertise into your own company and using other organizations. I think there's tried and tested experience out there that's available to tap into. And I think it's having the network to build it. There's lots of ways you can build that network.
Great, thank you all. We are a couple of minutes over, so I think it's unfortunately time to wrap up the discussion. But thank you all for all of your insights. It was a really fascinating talk. I think with cell and gene therapy, there's always so many dimensions. I think we covered a lot. So thank you so much for all of your points. But yes, I think with that, I'll hand over to Mari to wrap up.
Brilliant. Thank you, Shada. I'd like to reiterate that. Thank you to our speakers, Keith, Richard, Sarah, Susan, for all of your insight today. As Shada said, it's been fascinating. A reminder for the audience, this recording will be available on demand, so you can look out for an email in the next couple of days where you'll be able to access that once it is ready. But I'd also like to thank our sponsor, Lachman Consultants, for making this discussion possible. And on behalf of Lachman and Sightline, we hope that you all have a great remainder of the day. And thank you very much for watching and participating.